Hi everyone. Today we'll be going over PK submit. Here's a the today's webinar agenda. And by the way, just to start, I apologize if I'm getting through working through a cold right now. So if my voice cracks up, I apologize ahead of time. But without further ado, so this is what we'll be going over today. We'll do some introductions, kind of go over some common problems PK scientists run into, and then we'll go over PK submit and how that addresses some of those problems, and then some of the ways it can help the user with modifying their data so they can use it for analysis and generate these CDISC domains that they would need for submission. And then we'll finish it up with a Q&A at the end, like Sebastian mentioned. So just to introduce myself, my name is Michael Clifton. I'm the product owner for PK Submit. I've worked with Certara since 2020. I'm now product manager, specifically as a product owner for this product, like PK Submit. Before then, I worked as a pharmacokinetic research scientist for over 15 years using Phoenix Win on Len. That was I have a lot of experience using that. And as a PK scientist, I came across probably a lot of similar issues that many of you probably came to over the years, because I worked for a CRO. So when you work for a CRO and you're needing to work on these studies, you typically run into these same situations, right? You had these tight timelines for the submissions where you barely had any time to perform the analysis and review your results, especially when it came to studies like first to files, where you need to get those like right out the door. And then over the years, a lot of us have experienced more and more complex study designs, sometimes even combining multiple studies into one study, right? And then, when it comes to utilizing Phoenix, Phoenix is a very powerful tool, but it does, take time to train on it and try to figure out how to come up with these, using the objects within Phoenix to, modify the data in ways that we need for our analysis, and then, so you would end up having to create these like extremely complex workflows. That once you got the training down was very beneficial, but if you don't have that initial training or experience, it can be kind of difficult to get that down. And then another issue with just the data that we receive from outside sources for our analysis, it can be kind of dirty. You can get data from multiple sources, and then you have to combine that data in order to be able to use it and then format it for your needs. So this is where PK Submit comes in. So originally with PK Submit, it was designed for helping the user automatically generate these CDISC domains and supporting documentation relevant to non-compartmental analysis. And then those could be used for regulatory submissions, electronic submissions to regulatory agencies for the IND and NDA, right? But now with the latest version of PK Submit, we've redesigned it where we're still delivering those CDISC domains and submission packages, but now we're focusing on the PK scientists and how they would be prepared for their prepare the data for their analysis to be able to generate those CDISC domains for submission. So let's and the key points here is with PK Submit is we have a higher efficiency in NCA through automation, adding intuitive options that simplify the analysis process, as well as streamline the process for regulatory submission. So I want to kind of introduce you like a typical user workflow or scenario, as you will, for a day in the life of a PK scientist who receive data for their analysis that requires generation of these CDISC domains, right? So one of the first things we need to look into is data cleanup, right? So we import and prepare source data so that it can be used with PK submit. So how do we do that? Well, there's some of the main things that that we would do, especially with PK submit. We now have this single data model that we can map our source data to, and then that can be used throughout the PK submit process. So it makes it a lot. We map the variables that we were given to variables that are much more user friendly as we go through PK submit. And then if we have multiple sources, we can merge those files into one file that'll be used throughout the process. And then especially, if we need to make sure that the data that we were sent contains what we need, there's some data validation checks in place that can help with that. So now what I'd like to do is switch over to PK submit, just to kind of show you what we mean by that when we're cleaning up the data. So I have imported here, 2 data source files that we were sent. Now you can see here, these are similar to maybe CDISC domains that you were sent. But for PK submit, you don't have to use CDISC domains. You can use any source data set that you were sent. And we'll kind of show you how to map that and use that for PK submit. So if you look at PK submit here, the first step here would be going to the data preparation wizard. So when I click on that, first step would be we need to select our source data. So we have the EX and the PC files here, but now we need to assign those to a particular source type. So for the EX, that'd be our dosing information. So you can see here, we want to make sure that's mapped to the dosing information. And then PC, that's our concentration time data. So we need to make sure we map that to the PC data, PC concentration. And in doing so, it pre-populates mapped variables that we need for those particular data types. So you can see here, we have our result column. These are going to be the user-friendly variables that we'll see throughout the PK submit process. And then we have the source variables that are mapped to those. We can also adjust sample day and make adjustments to sample day and time for the analysis here to the right. We also have a preview of the data that we are bringing in at the bottom here. And you can see up at the top, this tab is for the concentration data, and then the next tab would be for the dose information. So it's the same thing here. You can make sure that the data here is mapped correctly and the correct merge keys are selected, as well as a preview of the dosing information at the bottom. So once we kind of have all of that mapped and adjusted the way we want, then the next step would be to merge. So we're applying a merge right now. And as you can see here, the merge was successful. We didn't run into any issues when running the merge. So that's great. And now if we can look at our final results here called merged worksheet. And this would be all of our, the source data and the concentration data and the dosing data merged together. And then we also have a log here where we can, verify what was merged and how it was merged. So you can see all of that set up here. And if you need to look at this log or any of this information here, you can also export that to your local directory. But I'm going to click on next because now we need to do a data validation check. So we kind of have these key validation checks in place to let us know if we're missing any important data like a subject number, a nominal time, analyte name, and units, and as you can see here, one of the main things that we're missing here is time units, right? So you can see that's missing. So you can either, go back to the person who's sending you the source data and let them know that, hey, we're missing this information. Can you send us an updated file? If you don't have that luxury, in the next step, we can show you how to modify the data so you do have that information. So next step would be submitting. And now you can see we have this merge result worksheet that's been created. And this is what we'll use moving forward to to perform modifications to the data, and then eventually the NCA analysis, right? So let me switch back to the slide here. So now the next step is data modification. So we need to modify and format the data for the NCA. So this could be, creating variables through transformations, duplicating samples. Say you're missing a predose for the next day's profile for a subject. You need to replace some values with the, based off of your BLQ rules. Or you might need to make some exclusions, like say removing trough samples from your analysis. So that's what we'll cover in this next part. So I go back to PK submit here. So the next step would be to use the analysis wizard. So this will take that merge result file that we created, and we can start making changes to the data. So the first place here is called column derivations and transformation. This is where we can, add expressions, where we can modify the data, create new data columns. So just to do a quick example of that, we were missing the time units for our analysis, right? So I need to modify that. I already know based off of the protocol and some other information that the time is in units of hour or H. So I'll just click the plus button here and enter time unit to match the variable that we have down below. And then I'll just add in single quotes H, and I'll hit execute. And you can see now that that's updated that variable, and we can now use this for our analysis. For any other more complicated expressions, there's a link here called syntax reference, and that'll take the user to a help page that'll kind of guide them through or give them information on how to write the expressions if they need more complicated expressions. And if that doesn't help, you can also reach out to Certara support for that. So now I click next and I go to the next screen here called analysis profiles. This is where we're basically setting up our NCA sort keys. As you can see here, by default, I already have the variables in place for our NCA. So I don't need to modify that too much. I do, however, need to verify that the data that we were sent is what we wanted. So how do I do that? Well, the data here is grouped by the sort keys. So you can see here for this analysis day 1, there's only one sample. So that's more than likely a trough sample. But then when you click on day 2, you can see, okay, for this subject, here's the day 2 profile. And you can see it's a full profile. So you get this kind of preview to kind of help you out with making sure that you have the correct NCA sort keys selected, right? And if, you can also set whether the study is serial or sparse. So there's a button down here called sampling design. So if you click on that, you can choose whether the study is serial or sparse. And then if it's sparse, you can also create an option here where you can combine the gender information. So you could have results for both combined gender or by gender separately. For this particular study, it's defaulted to serial, which is what we need. So I'm going to leave that as is. However, I do want to make sure I have my trough samples marked. So what I'm going to do here is you can use these filter options within the column variables header, and I'm going to filter on the ones that just have one sample. These are my trough samples. So what I'm going to do is select shift and then select the last row here, which will select all the rows. And then I'll right click and set all of these to trough, and you can see all of those are selected. And then I'll clear my filter, and then you can see that all of my trough samples have been marked. So once I've done that, then the next step would be to apply updates to the status codes and BLQ rules. You can see here we have 3 different result columns. We have a PK concentration, plots concentration, and display concentration. The PK concentration column is what we would use for as a source for our NCA. The plots, you would use that as a source for generating plots, and then the display concentration is what you would want to use for working on your report listings or anything like that. So for this particular example, I'm just going to use the first quantifiable substitution rule. I'm going to make sure my settings for the concentration, time, and LLOQ columns is set up correctly. And then I'm going to apply, so before condition, I want these 2 columns set to 0, and then the display set to VQL. And then after condition, I want those set to blank. And then for display, I want that also set to VQL. So if you can see here, once I select execute, you can see that these columns here were updated based off of the BLQ rules I applied. And that's another thing too. If you want to, as we're going, you can verify the data because there's this preview window at the bottom, and which also has the filter icon, so you can easily filter through data to make sure it's it's being modified properly as you go through the process. So we have our concentration columns formatted. Next step would be our dosing information. So we need to make sure for this particular study, we're setting the dose type. For this one, it's extravascular. So I'm going to just take this drop-down menu and select extravascular. If tau was needed, I could also manually enter in tau. If it was an IV study, you could also, enter in the infusion duration. But that's not the case here. So we're good to go, and we have the dosing information here. But if the dosing information, say, was incorrect, you can also manually modify the dosing here. So once we have that set up, then we can move on to the next step, which is where we can flag time deviations and create baseline concentration columns. I'll skip over that for now as I'm using nominal times for this demo. And now we're getting to a part where we need to duplicate samples. So as you could see here, for this particular example, the day 3 profiles are missing a pre-dose sample. So what I want to use is I want to use the last sample of a previous adjacent profile to create the pre-dose. So I've selected this rule, but you can see there are many different types of rules that could be used. But for this one, I want to use the last sample of the previous adjacent profile, and I want to apply this. So you can also select the concentration column you want this applied to. I want to apply this to the PK concentration, since that's what I'm going to be using for my NCA. So once you have those selected, you hit execute, and you can see in red here where we duplicated data. So you can see here it inserted this pre-dose sample for day 3. And if I scroll all the way to the right here and go to the PK concentrations, you can see here that it inserted the last sample, the 10.1 from the day 2 profile into as the pre-dose for day 3. So once we have that set up, then the next step would be to see if there are any concentrations we need to exclude, or possibly add any comments that need to be added to the CO domain for CDISC. Now, there are 3 ways to do exclusions. There's a manual exclusion here, where you can just right-click and select the type of exclusion or comment that you want to provide. There's a visualization where you can click on this visualization and then select a particular profile and subject and select a particular data point to remove for that particular subject. And then there's also conditional exclusions, which is what I'm about to demo. So we need to exclude trough samples. So I want to make sure those are excluded from the NCA. So I'll select exclude from NCA. I'm going to select the trough variable here. set that equal to true. So any samples marked as true for trough will get excluded. I'll enter my reason, exclude from analysis, and then I'll apply that. And you can see here, any samples that were marked as excluded are shown in red. And you know, another good thing about a lot of these different modules within the analysis wizard here is we can save our settings and use them for future use, especially for the exclusion. So if I want to make sure I can use the same criteria for future studies, I can save that criteria and make sure I can use that. It'll save that to your local directory, and then I can use that for future use. So now I'm going to hit next. And then this is where you can set your NCA settings. Make sure you have the correct concentration column and time column selected. You can also calculate partial areas, do unit conversions, carry along additional variables. Right. So once I have all of that done, I'll click submit. And now I've created a master concentration worksheet, which will then be used for creating the CDISC domains, as well as used in creating the NCA worksheets that we need. And you can see those are created right here. And as well as a master CO for the CO CDISC domain if we were going to generate one. So that's the analysis wizard. So that's you know, what we would use for data modifications. So now the next step would be finalizing our PK data. So we want to select our slopes for the lambda z criteria, run our NCA, and then pick the PK parameters we want to report, as well as the create any ratios we might want to report as well. And then this data will eventually be used as a source for CDISC. So I'll demo that next. So once I had finished creating these NCA worksheets through the analysis wizard, PK submit also generated an NCA object, and you can see it's labeled for plasma and then serial. And then it automatically loads those NCA worksheets. So this is already set up. All I really need to do is run it, and it's good to go using this information. But say I wanted to, I want to change the slope selections before I run the NCA. So the next step would be under PK analysis, you'd go to the slope selector, make sure you have the correct NCA object selected. And now we can go in and we can modify the slope selections for calculating lambda z. So you can see here, you just can click on each profile here, and it'll update the graph here. And it's very similar to the options available in the NCA. It might be it's a little easier to look at and use, but you can clearly, like, if I wanted to change this, I'll make click on that to make sure I only want to use these 3 points, and then you can see those points are displayed here under the select profile. You can also manually change these if you needed to, and then you can also enter in lambda z acceptance criteria. So I'm going to go ahead and select OK for the change I made. It'll make that change in the NCA object, and now all I need to do is just run the NCA. Okay, great. So now we have the NCA run, and now we just need to finalize the results. Finalize PK data. So this will go and grab the final parameters worksheet from the NCA that we ran. I'll select OK. Okay, so now this is where we will choose the PK parameters we want to include in our CDISC domains. So currently it's defaulted to these 5 main parameters, but we could include additional ones, say like Tmax, Tlast, for example. You can also save and load. If you have a same parameters that you're reporting every time, you can load those selected parameters in every time, using these buttons here. There's also, if you need ratios calculated, for example, accumulation ratio, you could easily do that here with this button. So you can see here for calculating the accumulation ratios, you need to select your ratio variable. So for example, analysis day, and it'll pre-populate the values that are under analysis day here, and then you can select which one is your reference value. And then you can select the prefix that is used for representing that ratio. So this one's accumulation ratio, but you could also select multiple other prefixes for your calculations. And then you can select the test values you want included for your accumulation ratio. And then you want to make sure you select the correct parameter you want reported for that ratio. So example, I want that applied to AUC last, so I select AUC last. I make sure I have the correct sort keys selected here. And then I want these calculated for individual subjects. You can also calculate mean values, as well as either doing a straight ratio or a percentage. But for now, I'm just going to do individual values. So I'm going to click add, and then you'll see it'll add the ratio here with the information that you used to calculate that. And then I'll select OK. And then you can see here it added the ratio here to the selected PK parameters. And now I'll select next. And now this is where we can do exclusions on the PK parameters. You could do conditional exclusions, for example, say AUC extrapolated, for anyone who had AUC extrapolated greater than 20%, I need to exclude AUC infinity. You can do that here. Same with R squared adjusted, you can apply those exclusions based off of those particular conditions. You can also do manual exclusions as well as adding a comment. So as you saw before, when I was making updates, you could see the master CO domain worksheet turned red. That's because it's being updated right now, in this PK parameter screen, because we could add comments here that would be added to the CO domain. So that's another good thing too, is whenever a worksheet becomes out of date, it'll turn red, and you can see that here displayed for master CO. So if I were to say just add a comment, test comment, and I click OK, you can see here it now added it here for where it'll be pre-populated for the CO CDISC domain. So I'm going to go ahead and click submit. So now we have our final results. We have the final PK parameters. So here it is in a stacked version. Now, as you can see here, it's actually listing all of the parameters generated from the NCA. When we generate the CDISC domains, it'll only use the parameters that you had selected. This is just to have all the information available in case you need it for your any other tables or listings that you need to generate for your reports. And we also provide a pivoted version of that here as well. And then you can see here the master CO domain was updated, so it's no longer red, it's up to date, and it has that comment that I added. So that is finalizing the PK parameters, the PK data. right? So now what we need to do is we need to generate or create our CDISC and validate those CDISC domains. So we'll go through the CDISC creation of the domains, submission packages, and then how Pinnacle 21 is integrated so we could validate those results. So I'll go through this briefly. So under CDISC, first step would be to create the CDISC domains. So here, we can select the standard SAS transport file and control terminology that we want to use. For this particular example, we'll just go with 3.0, but just keep in mind with PK Submit 2.0 and greater, we have the latest versions of send and SDTM. So you can see we have 3.1.1 for send and then 3.4 for SDTM. But for this demo, I'll use 3.0. And then for the control terminology, I'm just going to use the basic send terminology that we have here. So now I will select next. And now what it's doing, it's going to create this preview screen, kind of showing you the previews for the PC and PP domains that get generated, as well as the expressions that are used to create those domains. So here we have the PC domain. So to the right here, we have a preview of our master concentration worksheet and the data within that. We have the expressions to the left here that are used to create the PC domain file. And then we have the preview itself of the PC domain file right here. So based off of the standard version that you selected, some of these variables we can't change the expression because it's based off of the particular version of send or SDTM. But there are some variables here that can be manually modified by the user. So you can see here for PCCAT, instead of analyte, you might want to point that to a different variable. You can modify that. We're just going to leave that as is for now. If you did need to modify that, and you wanted to see those changes applied, once you modified it, you would click apply here and you can get that information to be displayed in this screen here. You can also update the terminology mapping if needed. But for now, we like how we have the PC set up. So I'm going to go to next, where now we need to modify the PP expressions if we need to. It's the same setup as before. We have a preview of the final PK parameters to the top right here, the expressions here on the left, and then a preview of the PP domain. And then, like I said before, there's some expressions here that can be modified. You can see here for this particular example, this is actually letting us know that some variables are, kind of incorrect based off of the expressions that we have in here. So, so you can see here for subject ID, it's letting us know that we can't find the column for that. Same with the site ID, right? So if you needed to modify those, you could update that to point to a variable that does have that information. Those two expressions aren't required for CDISC for this particular example, so we don't need to populate them. So I'll just leave that as is for now, and then we can continue. So once I hit submit, you can see here that it generates these CDISC output worksheets. And these are the ones associated for send. Since this was a serial study, we don't have a pool def domain created. If it was a sparse study, pool def domain would be created. And then if it was SDTM, we would also see the ATOM data sets created, ADPC, ADPP. So now the next step would be to generate the submission files from these worksheets. So we select create submission files, and now we can enter some information that'll be pre-populated in the define files, study description, protocol name, number, version number. I'm just gonna, just add some arbitrary names to this. And then once you select OK, now it's creating the define files, both in XML and in XSL versions of the define. as well as creating the XBT versions of these CDISC domains with the correct labeling. So you can see here under documents, we have the XBT files created, XML, and then an Excel version of the define. I also want to point out too, as we've been working on this PK submit workflow, we've been tracking our analysis through these log files here. So you can see with this log file, just to briefly touch on this, this has tracked all of the updates I've been making throughout the analysis. So you can see here, it entered in, that I entered in this expression for time units, the selections I made for BQL, and everything else. So you can see a lot of that information is being tracked and displayed within the log. Same goes for the merge log, what was done for the merge. And then we also generate a core output that is similar to the one generated from the NCA object, but this one captures the units. Because in some cases you might have data that has two different analytes with multiple types of units. So that is, we added this so that could be captured for the customer in case they need that for their reports. So we've created the submission files, and now what we need to do is we need to create the Pinnacle validation report and SDRG report. So this is based off of Pinnacle 21 community. It's very similar. Same situations where you can set up the engine, format of SAS, send, standard, configuration, all of that. And what this is just, and just to keep in mind too, this is optional for the PK submit user. They don't have to use this, but this is more of like a convenience for them. It's kind of built in, it's using your Pinnacle 21 community, and it's generating the report within the Phoenix project. So I have all my settings, and I'm going to go ahead and click OK. So once this is done, it will save the report to our documents folder, as you can see here. So we have the, this was a preclinical study, so this was an NSDRG. So we have that report created, and it creates kind of like a template. So the user will still have to put in a lot of information, but it does pre-populate a lot of the validated information that the user entered and that came from Pinnacle 21, so that gets automatically entered. And then under Excel, you can see we have the Pinnacle 21 report that was generated. And so I'll just briefly show that. So you can see here, it generated the report, and you can see here for the domains that we generated, there weren't any issues with this. So that, that looks good. And then once we have that, then the last step is just exporting all of the information to your local directory. So you can, and that's pretty much it. And then, I created this project through Integral, our Certara's data repository, and track that as an analysis within Integral. So actually, once I get to this point, I can go ahead and save my results back to the Integral data repository, and all that information will be stored there. And that is pretty much the demo of typical day in the life of a PK scientist working for a CRO, trying to get their analysis formatted and ready for use, and be able to generate those CDISC domains. So over the past few years, we've worked closely with some of the largest pharma companies in the world to help them streamline their NCA workflows. And PK Submit is the manifestation of all the learnings we gained through that process. So we were able to merge the capabilities into a single simplified user flow that I just demonstrated for you, that will help guide the scientists through key decisions, automate their decisions, recommendations, and exception detection, You know, as I showed you, it provided convenient tools that could be used, such as like the sample data duplication. You can also track the audit logs. I demonstrated that as well as the integration with Pinnacle 21 for validating your CDISC results. And as you can also see here, some customers might also be asking, you know, since the FDA, the CDISC has come up with 80 NCA domains. We don't have that available in PK Submit 2.0.1, but we are working on implementing that for PK Submit 2.1, which will be out later this year. And just to also include, you know, we didn't have much time to go over, but we had some additional capabilities I just wanted to list out that PK Submit could do. I briefly touched on, you know, the user could adjust the sample day and times. For example, you have a 24-hour sample that's marked as day 2, but you want to include that under day 1 for your analysis. That's where you could kind of make that modification there. That was done early on in the process during the data cleanup. We can also create dose normalized and baseline corrected concentrations. We can add time deviation flags, unit conversions. There's additional sample duplication rules we didn't really go over earlier, which can be based off of what you designate for tau, or what Cmin or CLAS is based on. There was a visualization. You could use that to exclude samples. It makes it a lot easier to see if you have, you know, a data point that's way off from the other ones in the profile. You know, there's a lot more we can, you know, show you with what you could do with the ratio calculations. You know, and then additional things with modifying the variables for the CDISC domain, PC and PP. I showed you that you could do that. There's, you know, much more, you know, to do with that if you ever needed to. You know, and there was also the fact that you could save and load the settings for each of those modules within the different parts of PK Submit, and then also those audit logs. And we can go into detail for future webinars if you want. Basically, all of these topics here could be their own webinar and demo to really dig into if you have questions with any of those. But that is it for the PK Submit webinar. I guess we'll leave it up to the Q&A now. Sebastian, do we have any questions?
Yeah, thank you, Michael. Yeah, great input. Thank you very much. And we have indeed 2 questions that came from the audience. Again, if you want to ask your question to Michael, please use the chat, and I will go ahead with the first question. That is, do I need Phoenix and Win-on-Lin in order to have PK Submit?
It's a great question. Yes. So PK Submit, it's a plugin within Phoenix that utilizes the Win-on-Lin NCA object, as well as some of those kind of basic tools within Phoenix. So yes, you would have to use it since it is a plugin for Phoenix.
All right, thank you. Another question came through, and that is, can you go back in the tool and correct an error?
Yes, so I can demonstrate that right now. So say, for example, I needed to modify the dosing information or one of the expressions in my column transformations. So what you could do here, when you click on the analysis wizard, you get this message here that asks you if you either want to reset the analysis using the latest merge results. For this particular example, I don't, because the merge results that I'm using is fine as is. So I do not want to click on yes. That would only be the case if, say, we received a new data set, you know, that updated the concentration time data. If that was the case, we would probably have to go through the data prep wizard and recreate a new merge result file. But since our merge result file is okay as is, I just need to modify anything that was done in the analysis wizard. So in this case, I would just select no, and it will bring up the analysis wizard. And you can see here that we now have these active links to the different sections. So say I needed to update the dosing information. I could click on dosing here and it'll take me back to the screen, and I can, say that this dosing, it needed to be 150 for whatever reason, or I forgot to enter in the tau information. I could do that. And then you would just simply go through, keep clicking on next, because it's also remembering all of your settings that you did before, as you can see here. So I don't need to repeat any other steps. I just keep clicking next until I get to the submit button. And then when I click submit, you'll see that now all of this data is out of date, and that's because we have now updated our master concentration worksheet as well as our NCA results. And if and now we just need to repeat our the rest of the work in this regards. So we would need to rerun the NCA, go through the workflow for finalizing the PK data and the CDISC, and once you repeat all of those steps, then all of these worksheets will go back to being black, which will mean it'll be up to date.
Thank you very much. Another question came through. Can PK submit be used to create all CDISC datasets?
No. So PK submit, its main focus is in creating any of the CDISC domains that are associated with the PK analysis. So that would be the PC, PP, RailRec, sub-PC, CO, pool def, and then any of the ADAM data sets, ADPC, ADPP, and then in a future version of PK submit, we'll be able to do ADNCA.
Okay, perfect. Thank you, Michael. Yeah, thank you for the question. I don't see any more questions that came through. But please feel free to reach out to us if you have further questions. We are going. Oh no, there's one more question. I see. Then I think we have time to cover this.
Sure.
Would it be possible to merge data from the two different sources? For example, data from Excel spreadsheet and data from Watson.
Yes, that should be fine as long as both data sets are brought into the data folder here. And then you'll just simply, when you're running the data prep wizard, you'll just, need to make sure you're selecting the correct source information for that. So if they are both concentration data, you might be able, to select both both of those as that particular source type, and then make sure that this is selected, and then you would and then in the next screen, obviously you need to make sure that the mapping is appropriate, as well as the sample merge key, because you'll need to make sure that you're merging on a common variable between all of the data sets in order to get them to merge properly.
All right, thank you, Michael. If you have any further questions, yeah, feel free to reach out to us after the webinar at any time. We will cover these questions.
Oh, and Sebastian, can I add one more thing? Yeah, and I just want to make it clear about the the Pinnacle 21 option that's available in PK Submit. I just want to reiterate that uses Pinnacle 21 Community. If you're a user that uses Pinnacle 21 Enterprise, you can still use that, but that would be outside of Phoenix and PK Submit, which should be fine. Like I said, you can take once you've exported all of your data sets to your local directory, then you can use Pinnacle 21 Enterprise like you normally would. It's just that Enterprise does not work with PK Submit. It's only Pinnacle 21 Community. So I just wanted to make sure that was clear.
All right. Yeah. Thank you for clarification. I think we can wrap it up for today. Again, we will send you the recording from today's session, and feel free to go back to rewatch it, to share it with your colleagues. And if you have any further questions, feel free to reach out to us. But other than that, I think thank you, Michael, again for that great presentation. Thank you for your engagement, dear attendees, and see you next time in one of our next webinars. Take care and goodbye for today. Thank you.
Great. Thanks, Sebastian, and thanks, everyone. Have a great day.